if I already started to go dreaming, the next level is to analyze single mitochondria, because within a single cell, there are many, many mitochondria. And what we already see now, I guarantee you, there are mitochondrial subpopulation. So not all mitochondria within a single cell are the same.
Welcome back, everybody. We are joined today by Gena Nikitin, the founder and CEO of Miphic. Miphic is trying to improve mitochondrial medicine for people. Is that correct?
Yes, this is absolutely correct.
That's going to be super cool for people who probably heard about mitochondria a lot in high school, as I did, the powerhouse of the cell, they used to call it. So it's responsible for a whole lot of really important things that go on in the body. And my understanding, it's a very untapped medical market. It's been very difficult for anyone to make any sort of headway in that space. So we're excited to kind of learn how you've been improving that and what your goals are in the future to make this a much more accessible and useful field of medical study.
Yes, absolutely. Thank you so much for having me. I'll be happy to talk about Miphic, talk about our mission, how we're going to improve medicine, hopefully worldwide for everyone. That's definitely our goal. And yes, about mitochondria, which is my personal passion.
Super cool. So I'm curious, it's a kind of a specific field to be getting into. So how did you end up here in this field? Have you always been interested in sort of cellular biology, or was it something in your past that kind of got you here?
Yeah, so it all started actually back in my childhood. So yeah, I had a really rough childhood, so really rough upbringing. So from the earliest days, I actually wanted to make something that is good for humanity, so that it actually compensate for what I saw during the childhood. And from the very early age, I decided to become a scientist, because that would be my way to move the humanity forward, to advance it. So initially, I actually didn't know what I wanted to do in science. But then about 12 years ago, in the beginning of my PhD in Switzerland, I identified a problem which, if I would have been able to contribute to being solved, that would actually mean that my life would matter, and that would actually fulfill my childhood dream to contribute positively to humanity. And that was that we ignore mitochondrial function in clinics, while actually they are super important for everything. And then I quickly realized why we ignore them, that the technology which gives access to mitochondria in real life clinical patients is just not there. So basically our technologies are not good enough for the real world. And to begin with, I started to try to optimize everything that existed already in mitochondrial research. And then I realized nothing really works, so that it's actually can be reproducible, reliable, and I started to develop my own technology. So it took me overall pretty much a decade, 10 years, to get to the point when it was mature enough so that I could start company like about 2 years ago and start bringing it to the real world, which we're doing now with Miphic.
Yeah, when we first met, it was through one of the Slack communities, and I had posted that I was dabbling in biohacking and taking an epigenetics test to get my biological age, and I was sort of underwhelmed with the results that I had. And so then you reached out to me and said, hey, let's talk about this. So maybe for the people listening, if you wouldn't mind kind of repeating the conversation you and I already had, where what is the difference between what people are doing today with the epigenetic testing and then what you're doing?
Yep. So Miphic one-of-a-kind approach is that we focus on function. Function is the final product of all of the possible causes. For example, genetics, epigenetics, the particular nutrition of a person, and so forth. But ultimately, what determines how well mitochondria work right now in every particular cell in a body is the combination of all of these factors. And if mitochondria, for example, would stop working completely, we would be dead within microsecond or something. And if they actually are corrected in their work, so the final functional outcome would be also much better for a patient, for a human being, for the quality of life, regardless whether or not the genetic factor is good or bad, whether or not the epigenetic risk is higher or lower. Right now, today, for example, why do you feel tired or energized? To a big extent, it might be that your nutrient supply to your energetic systems, in particular mitochondria, right now either optimal or suboptimal. So, for example, we know that metabolites really accumulate. So the waste product of metabolism is being overwhelming for the cellular systems right after the training. That's why we need to recover. And that's actually an example of function. While genetics actually is exactly the same, or epigenetics pretty much is exactly the same before your training session and after your training session, for example. However, as we probably all understand, we are not the same right before the training and right after the training. And that is function. That's what we actually measure. And mitochondrial metabolism would be immediately telling what's going on with the actual capacity in the cellular, metabolism pathways right now, like this microsecond. And we could use this information to actually improve longevity, improve the quality of life for the patients, and also hopefully cure very serious diseases, or at least prolong the survival during the very serious diseases. That's our goal.
And I specifically remember you saying that with the epigenetics testing, it's harder to influence. And with your mitochondria research, like there's going to be options that can help improve the function. Can you talk a little bit about that?
Yes, absolutely. Yes, because we so far really are not even being close to altered genetics in a clinical setting in any basically reliable or straightforward way. However, we already know that targeting mitochondria has proven to be one of the best approaches to particular types of blood cancer, for example, acute myeloid leukemia. This is a particular focus for us. This is our entry point to bring our technology to the market. And in this, for example, particular cancer type, which is very deadly, there was no survival improvements for almost half a century, except that during the recent few years, the survival was improved on average by drugs which target mitochondria. So we already know that this approach has great potential, at least in certain types of diseases. However, the problem there is still that it doesn't work for all patients. The patients have the same disease, same diagnosis. However, up to 60% of patients are non-responders to targeting of mitochondria. So our technology, and this is where we hope to bring unique value for the patients and go basically and be the first on the market of mitochondrial precision medicine, is to measure how exactly mitochondria of cancer cells in a particular patients are different when they respond to a mitochondria targeting drug or not. And we already have some proof of concept that we could do this. We are now just trying to scale this technology.
That's really cool. I'm curious to know a bit more about where the previous technology fell short in being able to either measure or influence mitochondria, and how you are improving that. Obviously, you don't have to go into company IP level of detail, but I'm just curious to know what the shortcomings were and how you're trying to address those to make this more effective and useful area.
Yeah, this is an excellent question, and that's in fact where I spent pretty much 6 to 7 years of trying to find and trying to get to this technology which we're using in Miphic. And that's basically to satisfy two criteria. First is that if you measure something, then you cannot throw away the data and basically say, oh, I don't like how the data looks, I'm just not going to use it. Because this is scientific integrity, and that's what actually you have to follow in the real life, because you cannot ignore in the real clinical setting if you don't like the data. And the second criterion is that if you have certain data which is measured by a technology, then you should be able to reproduce it within the statistical error. If you do the same thing, the same experiment, or the same test in the same patient, it has to be reproducible within the statistical error, because that means that your methodology is, or technology is, reproducible and can be used to reliably help a patient. Turned out that all of the previous technology do not satisfy one of these criterion. Either they rely on people use data selectively, or they cannot reproduce their own data. That's what Miphic is all about. It was years and years of developing technology which is reliable and reproducible. Then we could bring it to clinics.
Is there any particular reason why it wasn't reproducible the old ways?
Yes, the previous technologies, roughly speaking, if we simplify things, bulk technology, meaning that they measure total signal from all of the cells, meaning that if during the analysis you have, let's say, 1,000 cells, you would get 1,000 times higher signal than if you have one cell. And in reality, it's almost impossible in practice to have exactly the same amount of cells, exact amount of mitochondria within the cells all the time. So technology must be internally normalized, and this is what Miphic delivers. It doesn't really matter whether we are measuring 2 cells or 2 million cells. The technology is on a molecular level, internally normalized. This is what is the unique know-how. I'm not going to go too much to technical details, but yeah, that's how it works. And this is what is the unique differentiating factor in comparison to all of the previous technologies, I would say.
Yeah, that makes a ton of sense. I'm kind of curious, like, how far have you, I don't know what stage of development of Miphic you're in, but do you have a plan for how you envision patients taking your test and getting their results? And like, what is the experience going to be like from the end user's perspective?
Yes. So, of course, times are super rough now. Investment climate is not the best, let's put it mildly. But if we are successful in following our basic general baseline roadmap, we are planning to enter the US market in acute myeloid leukemia as the companion diagnostics, basically when we will be taking the mitochondrial profile of a patient and recommending that certain drug should be recommended for that patient or not, with a very high precision of prediction that this drug would work for that patient. So we are planning to enter the US market in 2027. So in, let's say, 2.5-3 years from now, we hopefully bringing this to the real world for a particular rare blood cancer, which is extremely deadly right now, and where mitochondria targeting proved to be the first ever breakthrough in survival for these patients, but only for a subpopulation of these patients. And we want patients really be sure that the drug that they are given is truly helping them, instead of giving them only side effects. Yeah.
So you envision this working alongside, like, epigenetics is sort of in that B to C space, but that doesn't seem like Miphic is going for the same strategy here, because, you know, I'm not sure if I got this right, but if you have people being treated for cancer, like, that's going to be in the context of a lab with a doctor, and it's more than like a at-home blood test, right?
Yes, absolutely. Well, because to be honest, what I am particularly interested in is to try to revolutionize the medicine, and that can be done if we solve a really big pain in medicine. For example, that patients are getting a very expensive drug, and there's up to 60% chance for some of them that this doesn't work, only gives the patient side effects, and there's no way of knowing right now why and who would be responder. That's the whole concept of precision medicine, and we are going to be doing it from mitochondrial perspective. But maybe, yes, to answer your question, Anthony, definitely we are super interested in, more of a lifestyle type of mitochondrial approaches. In fact, we do have a lot of people from longevity space and anti-aging space contacting us in terms of, trying to do something for them. I cannot talk details because, I have NDAs and so forth. But definitely we are certainly very interested in this space as well. Since this is so far rather, let's say, side projects for us, I'm not considering this to be a main go-to-market strategy. But of course, if suddenly this demand explodes, which so far did not happen, we only have the first forays into that space, definitely we're going to be focusing much more on this, let's say, B2C, business to customer space in terms of mitochondrial screening as well. And by the way, I very much welcome anyone who is interested in this, contact us, talk to us, because we definitely are welcoming any ideas, any potential, side projects, because we can, if you have the samples, which you can send to us, like samples of live cells, and you are interested in a particular mitochondrial question, we definitely can work with you. We are very cost efficient, we are very fast, so totally open to ideas. Yep.
Yeah, I know. I for one am totally into this and like to play and see what kind of cool things I learn along the way. So we can talk about that offline for sure.
Sort of along those lines, I'm curious to know about if there is value in regular like mitochondrial examinations for yourself, so you can see just generalized health trends and use those as indicators, like early indicators when something might be going in the wrong direction.
Absolutely. Exactly. This is what actually touches what we have just discussed, and especially we're interested in aging problems, because what we definitely can be sure of is that mitochondria in babies are extremely more efficient and work extremely and are extremely more powerful and work much better than mitochondria of elderly people. And we're sure that there are ways to push mitochondria of basically an old phenotype towards a younger one. Yes, so far these are actually first forays which we are having, and again, there is not such a big focus on it right now, because as a startup we are focusing on our main go-to-market strategy, which is deadly blood cancers. But if we have customers, and it seems that we do have customer demand, we definitely can do this as well. The easiest is aging, and then of course we hope to do the following. We basically screen mitochondria in, let's say, 5,000 patients, and someone provides us their lifestyle data, clinical data, who has diabetes, who has obesity, who had a heart attack history or has a high risk of heart attack genetically. And then we see, we train machine learning models, and then when we see what type of mitochondrial profile is predictive towards one of these lifestyle outcomes, not necessarily clinical outcomes. For example, high risk of type 2 diabetes or obesity. And then we also, as the next step, compare how this mitochondrial profile is different in which aspects from the healthy people. And this is a good hypothesis to therapeutically now push mitochondria from the more problematic phenotype, so the way they work, towards the more healthy behavior. And then we see if we can actually come up with a new anti-obesity drug, generally new longevity supplement and so forth. That's all of, this is so interesting for me and I'm so passionate about this. So I'm being torn, basically into two different directions. One is scientist and enthusiastic person, who is super, hyped about mitochondria. And on the other side, I'm a CEO of the company. I have business demand, focus, go-to-market strategy, and I have to put the priority towards that just for all of us, for people of Earth, essentially, to get mitochondrial precision medicine as soon as possible there. Miphic has to survive from the business point of view, become successful, then we can expand. So to summarize, I see Miphic as the first introducer of mitochondrial precision medicine into the world, and then the world picks it up, and who knows how mitochondria would be used in the real world, in longevity space, in diabetes space. That's my dream. No one company, not even talking about one particular group of people, can even imagine how mitochondrial function could be manipulated, corrected to help human health, and in which areas. It's like, this is what I like as a comparison. It's like introducing the internet for the first time and asking what would be the best implementation of internet in the real world. To me, that's what would be the best mitochondrial function, target, what would be the best area to target mitochondrial function in terms of diseases or general lifestyle, health, and so forth. Who knows? This is my dream. I just want this to become routine, reliable methodology, and then doctors can use it how they want. Patients, the world basically starts playing with it. And then I see that I did that contribution to humanity, which I dream of. And that's my lifelong work is actually becoming fulfilling for me. Yeah.
That's awesome. Like, really, really admirable. And I think what you basically, like, if I heard that right, it sort of sounds like you want to be the catalyst that unlocks, like, you're building the core platform, and then what people do with it is anyone's guess, but you've unlocked a new capability that we don't quite yet have. So if we can switch, I want to switch gears just a little bit here. So you're talking to a couple of tech nerds, and this is it's like a pretty, you're talking about machine learning, and it's going to be, I imagine, a very complicated machine learning model. Can you talk about some of the technical, like some of the cool things, and then some of the challenges that you're facing with with technology, and just sort of how you're working through that?
Yes, I would say one of the biggest breakthroughs for Miphic is connected with actually recent years and developments in overall AI space, so in particular computer vision. So Miphic technology is physically based on acquiring lots of microscopic images of mitochondria. So we have unique technology which basically puts some fluorescent dye inside mitochondria, and we know how to do many biological tests with mitochondria using this dye. But from a technological point of view, we have essentially lots and lots of images of mitochondria, of many cells. And so one thing is that we have so much information that's just overwhelming. So we now need to get this unstructured imaging data towards the structured, quantifiable, basically parametric data, which we can then use to train the models. And that can only be done by certain image processing algorithms. This is what we also develop. And I would say one of the biggest advantages of Miphic is that we, for the first time ever, can do this now on a single cell level. So imagine basically that we have like hundreds and thousands of cells in our images, and what we know extremely well, that different cells actually are not the same. So even within the same patients cancer cells are actually extremely heterogeneous in terms of how they work so how their mitochondria work, and to be able now to start really understanding what is the proportion of cancer cells which use this type of mitochondrial metabolism versus that can only be done analyzing each cell separately. And for that, with our microscopy imaging approach, we could do, with the help now of computer vision, so with the eye, segmentation of single cells and analysis of different cells. And yeah, I probably now really, again, forgetting myself and getting super nerdy and super technical. But let me give you rather actually one of the most important potential applications in the real world, which we hope to achieve this way. And that is identifying cancer stem cells. So very rare subpopulation of cancer stem cells which survive the general therapy. Imagine like you kill every cancer cell except from 1 out of 10,000 or 100,000. But then this cell survives the initial therapy. In a few months, it multiplies, evolves, and boom, you get a relapse of cancer in a patient, and now it doesn't respond to the previous therapy. So one of the potential useful applications of the single cell resolution is that we will be able to identify these cancer stem cells in the future and find the therapeutics which would hit specifically them so that they don't survive. And hopefully we could start moving towards the complete cure of cancer at some point. That is my dream. And there could be so many things where it could also be useful for. For example, for lifestyle, general health approaches, we could just analyze different types of blood cells in healthy people or semi-healthy people, T cells, immune cells, or some other type of cells, and then see how their profile separately is now changing during the lifetime of a person, and what is actually now connected to the risk of diabetes, heart attack, and so forth, how to catch it early, how to target therapeutically to protect people, to expand longevity and so forth. So I have so many basically potential ideas what it could be used to. The most important is that we now have to tune once again that technology, make it super robust in terms of how we analyze single cell mitochondrial profile every time. Then again, the world can use it. We basically introduce it to the world. It revolutionizes again basically the medical knowledge which we get from a simple blood test. And if I already started to go dreaming, the next level is to analyze single mitochondria, because within a single cell there are many mitochondria. And what we already see now, I guarantee you, there are mitochondrial subpopulation. So not all mitochondria within a single cell are the same. I mean, it's a little bit, going to a very far future, but definitely in a far future, there is no reason why we shouldn't go as granular and as precise in analyzing how the cells work, how it's connected to the self, as on a single mitochondrial level. That's basically how I see the future of the technology for Miphic. And again, only the world will know what kind of benefits it will bring to all of us in terms of healthcare.
I'm going to say, I love watching you talk about that because I can just see how excited you are and the passion behind it. And it's really, really energizing. So, hats off to you for finding something that really gets you this excited and is incredibly meaningful.
Thank you.
So one of the last questions I think I have is, what are some of the goals that you have that we might be able to help you attain in some way or another?
Well, for sure, one of the biggest asks from Miphic would be if you guys can spread a word, to the world, to the people that, look, we do have the technology which basically can be now used to totally scale mitochondrial profiling for whatever application, so that people who are interested in that contact Miphic. We work together with them. We find the new cool approaches. We develop new therapeutics with new pharma companies. We basically develop new diagnostic tests with doctors, so that we all work together and all bring it faster to the real world, especially because definitely the US is the market where magic happens. I mean, that would be great. So just spread the word and yeah, let the world decide where everything goes.
Yes, we can certainly do that. I've been very vocal about my research, my personal research, what I've been doing. I share it with all. Like I shared the results with Jason on the biological age, and so happy to do so is my point.
Gena, thank you so much for taking the time with us today. It was very interesting, very exciting. I wish you nothing but success in the future. This is super meaningful work, as I said before, and I really hope that you make some really cool breakthroughs and change the world.
Thank you so much, Jason. Thank you so much, Anthony. Great to talk to you guys, and yeah, good luck to everyone. Good luck to our audience. Yeah, and let us all have healthy and and powerful mitochondria. Good luck.
Be well, everybody.